challenge is solely focused on the accuracy of our methodologies. What is the best way to find mutations in cancer? We don't care about how fast they are or what are the computational efficiency or even how user-friendly it is. Those are really important things, but at the moment we just don't know how good these methods are and which are the ones that we should work on speeding up, optimizing, making more user-friendly. And so we designed a challenge that had two parts, meant to try to address that in good detail. The first part involves, or I should say the second part chronologically, involves real tumor data. We had selected 10 tumor normal pairs for reasons that Lincoln just mentioned. They're ICGC tumor normal pairs. They were whole genome sequenced to 50x in the tumor, 30x in the normal. They were selected from 2 different tumor types. 5 were pancreatic tumors. These tumors have very different characteristics in terms of their copy number and mutation profiles from the other 5, which are prostate cancers, which have low somatic mutation rates at the point mutation level. We made the raw and the pre-processed data available. We gave people our initial assessment of what we thought a good alignment pipeline would be, but we also made the raw FASTQ files available for groups to look at. And we made everything available. We tried to make all the clinical information that we had, and sometimes people ask questions we didn't expect. Somebody asked how old the patients were. I don't know what they did with that data, but we made the patient ages available, and we tried to make it as much as possible like if you had a series of sequences from patients at your own institute, how would you analyze it? What data would you actually know? And something like patient age would be readily available. Ethics approval was occasionally painful. Of course, it's personal health information, so we needed to get an ICGC-DACO application for each individual. We eventually sought and received an opinion from the Western IRB before we started to allow groups to access the data to provide some support for countries that had different ideas about how PHI should be regulated, and of course the regulations are very different. And we provided a template form to try to expedite a lot of the approvals at different levels. In the end, we had about 400 people who went through that process, so we know that it was something that was viable, but at the same time, we also wanted to make sure that there were data sets that were more easily accessible. So working with Adam Ewing, who was then with David Hausler at UCSC, but is now at UQ in Australia, we developed a tool called BAMSurgeon for simulating tumor data. The basic idea is that we'd start off with a genome, a cell line, for example, and at the read level, we would insert new SNVs and structural variants. And this tool allows you to basically insert them directly into the reads. We'd insert an initial group to create a normal-like that was not exactly like the individual cell line, and then an additional set for somatic variations. And that allowed you to create an artificial or a synthetic tumor normal pair, something that the exact truth would be prior known, that everybody would be able to agree on exactly what it was because, of course, we spiked it in. And we had 5 releases. I don't know why this says the 3rd is active now, but that's totally untrue. And we made them of increasing complexity. So the first release was very simple. It was 100% cellular tumor genome, no complexity to it. By the time that we got to the 4th, there was significant normal contamination. There were tumor subpopulations at different quantities, and we tried to make this a much more difficult and realistic-like problem, but with the idea that at the beginning this would draw people into the field who maybe had never worked on this problem before. So this is a quick overview of BAMSurgeon. The idea is that it will insert mutations directly into the reads and then realign those reads to figure out where they might have mapped otherwise to make sure that we don't have some sort of an artifact in that way. The data was made available through a number of different mechanisms. So, of course, Synapse was the main place for submitting results and for the leaderboards, but Anai generously donated download capacity through their GeneTorrent software. And then there was a cloud-based option where groups could directly access the data in the Google Compute Engine, and Google provided some free computing credits for it, or they could download it through Google. And I'll show you the kind of numbers for that in a couple of minutes. So in total, the challenge was structured as having the human tumor data and the simulated tumor data. These are challenges 1 and 2, and within each, we asked groups to try to identify structural variants and single nucleotide variants, and similarly for each of the synthetic tumors and each of the single nucleotide variants and structural variants for each of the synthetic tumors. So the challenge scoring occurs in two ways. For the real tumor normal pairs, this actually portion of the challenge is almost finished. Many thousands of candidates, actually it's probably going to be about 20K now, are going to be validated by deep resequencing with an orthogonal technology. So we're using a different sequencing technology that has a different error profile. We're doing targeted capture resequencing to identify breakpoints for structural variants and single nucleotide variants by identifying them in the population. And the resequencing will be quite deep, at least 300x, but probably closer to 500x for many of these, allowing us to make sure that if a group is identifying a mutation that's only present at 20%, that we don't erroneously deem that to be a false positive. By contrast, for the synthetic tumor normals, we know exactly what the ground truth is. In fact, the ground truth was made available as soon as each synthetic tumor finished, and we calculate sensitivity, specificity, balanced accuracy, and the F statistic, or F score, I should say. And so this is the rough timeline of the challenge, and you can see that we're about a week away from the close of the real tumor challenge, and then we expect to have a 2-3 month period for experimental wet lab validation, letting the winners know in February and announcing them at the SAGE Congress in April. So to date, there are 373 registrants to the challenge. On the 4 in silico tumors where there is no ethics approval required, there was 1,037 entries to date. And on the 10 real genomes, as of a day ago, it was 1139. It's actually up by 50 or 60 since then, and counting. And maybe one of the most interesting things about this is that there's an ongoing increase in submissions to the leaderboard. It was a question that came up this morning. We intend to keep the leaderboards for these open kind of in perpetuity so that it'll provide a community resource that people can benchmark new algorithms against. And in each of the 4 closed in silico genomes, we've seen that. There are dozens of additional submissions with groups trying new parameters and new tools. It's also led to a lot of discussion on the right way to score structural variants and how to really understand the ways in which we can simulate tumor genomes. And the simulator itself has greatly improved over the course of the challenge itself. So this is kind of a progress of the challenge, starting off way back when we announced this a year ago at the DREAM conference last November in Toronto. And you can see that there's been a gradual increase in the number, not that one, gradual increase in the number of people submitting results. And you can see a really striking peak in the last week. This is the close to the deadline of the Real Tumor Challenge, and 1,000 submissions came in within like 5 days, which is really kind of humorous. You can also see that there's downloads happening directly through Google and downloads happening through Anai, and then a pretty steady rate of people accessing the data through the web. And what's really interesting is that actually, even very recently, like 3 or 4 weeks ago, people were still downloading the data sets. And initially I thought these were late registrants who were downloading the real tumors. Actually, these are people who are downloading the in silico data sets and want to use this as a benchmarking to improve algorithm development. So we think that this will create a very useful gold standard. I'll show you some summary results of some of the major figures. So this is the first in silico tumor, and this is a summary of the SNV submissions. And you can see there's a pretty interesting range here of submissions ranging between recall and precision, but in some sense, all the groups do fairly well. I mean, even this group here at the bottom, sure, they don't have great precision, but they recalled everything, and they kind of made a different trade-off. And if you zoom into this fraction here, you can see that there's a lot of clustering of the best groups together, and you can see a little bit of deviation. That's about an 85% sensitivity and precision and recall to about a 95%, but there's not a huge amount of variability between those. What's interesting, though, is that the groups got there in a very surprising way. What you're looking at is the parameterization order for these groups. So the blue bars or the colored bars along the top reflect different groups, and these are their submissions over time. So for example, this is the winning group, and their first submission was quite good, but after they got the leaderboard feedback, which used a small fraction of the data for the leaderboard, they were immediately able to improve their results by a significant margin. That's a fairly common feature where most groups were able to do so. And it actually suggests we might think about genomic sequencing analysis in a different way. Instead of going forward and sequencing things, doing the analysis with a single parameter set, we do a small amount of validation and use that to optimize the parameter set, maybe even on a per tumor basis. That turns out, that general phenomenon turns to be reproduced in many additional tumors, all 4 of the in silico tumors, and I'll show you in a couple of minutes in the structural variants as well. One immediate concern that we had was that this reflected overfitting, that groups were using the leaderboard data to overfit the results very strongly. And what you're seeing here is the F-score on the testing and the training data set, and you can see it's tightly clustered, and if we look at it over time for individual submissions, you can see that groups aren't really overfitting on any individual metric or on the overall F-score. Instead, the training data set is providing useful information, but not in a way that doesn't generalize to the entire genome. And likely this reflects the fact that the human genome is so large that even 10 or 20% of it gives a huge amount of information. Observation is reproduced in all the individual tumors where, for example, this is in silico tumor 3, you see the same very strong correlation, and again, no significant increase in overfitting as groups parameterize. We looked at the classic wisdom of the crowds question, asking if an ensemble of different groups would do better than any individual group. You can see this is the single best group, and these are ranked in order of individual submissions, and you can see that there are several ensembles that do much better than a single group by themselves, suggesting that we might want to pursue this idea of merging multiple pipelines together despite the computational expense of it. We noticed that there were clear error differences in error profiles across regions. So the coding region of the genome had the lowest rate of error. By contrast, the intergenic regions had the highest error rates. It suggests that groups had previously optimized their algorithms on the coding region and that there hadn't been large data sets available to allow optimization on intergenic regions. Even within the coding region, on every single tumor that we've assessed to date, groups did not do perfectly on it. So in the first in silico tumor, the first 2 groups were perfect in the coding region of the genome. Every one of the other submissions had at least one false positive or false negative in what you might have imagined to be the clinically actionable or most likely to be clinically actionable region of the genome. Maybe the most surprising finding that we had was taking a look at the false positives and how they were phrased within the genome. And one thing that we looked at was the trinucleotide profile of the false positives. So for example, these are C to A mutations, and this gives the nucleotide context, the 5' and 3' base relative to it. And we found that there were a very, very striking pattern of trinucleotide variation. It's a deamination profile, and it's preferentially present in the false positives. In fact, it's preferentially present in the false positives of all of the tumors that we've looked at. And this strikes us as likely to be very interesting because this is the profile that was identified in a study last year as the most common trinucleotide mutational profile in human cancers. So it's a deamination profile which could occur for a lot of reasons, but one of them appears to be straight false positives because of low read frequencies that are present as noise in the sequencing experiments. We looked at the structural variation submissions as well, and you can immediately see a huge amount of scatter. So this is the first in silico tumor, the one that we tried to make the easiest, and you can see no group did better than about 85%, the highest recall. Many groups had very high precision, but a lot of groups, even the best submission from several groups, wasn't actually a great submission, and it really reflects the challenge of this problem. Again, there were clear parameterization benefits from groups managing to greatly improve using the leaderboard statistics, although, and this is again consistent across tumors where you see huge scatter across methods and significant parameterization benefits. That was in silico 2, this is in silico 3. But what's surprising is that there is a little bit of overfitting. Actually, it's the other way around. There's actually a little bit of underfitting. So for whatever reason, the training data sets that we picked for the first 2 in silico tumors for structural variants were a little too hard, and the rest of the genome was actually a little bit easier. So groups tended to do somewhat better on the entire genome than they did on the training data set. So let me summarize the results that we have to date. The first is that there's these surprising trends in error profiles. I've mentioned the trinucleotide bias. One thing that appears to be coming up is that normal coverage is much more important than we had anticipated, and the typical 30x coverage that groups have been using is probably too low. In bases that had higher coverage, there was more accurate somatic mutation detection. It also very clearly highlights the importance of parameterization, and I think that's something that we'll hear about a little bit in the talks that follow from the winners. And it's nice to see that there's the evolution of a community around the idea of rapid algorithm development and benchmarking, which I think is going to be a long-standing benefit of this challenge. And in particular, we'll see how the winners of the real tumors compared to the performance of algorithms on the in silico tumors, and is using in silico tumors an appropriate methodology for benchmarking and doing algorithm development in the future, rather than having to go to the difficulties of distributing real human tumor data. And so what comes next? A couple of things are ongoing with this challenge. The first is that the Real Tumor Challenge closes in a week, I guess. Then there's a round of experimental validation that we think will take until around February. We're also going to release continued sets of in silico human tumors. And the idea here is to provide a technologically recent benchmark at all times. So the tumors that we benchmarked here were sequenced a year to a year and a half ago. So that means that they used, for example, protocols that involve PCR. There are no PCR protocols, and how well would those do? And so we'll continue to release these, but with much longer timelines. They'll be released every 6 months, giving groups a chance to look at them, not be quite so rushed in making the submissions, and they will be trying to update to look at important technological shifts in read length or other sequencing technology aspects. In February, we're going to reopen the leaderboards for the existing challenges. This is not to change the winners in any way. Those will be based on the leaderboards as they stand a week from now. But we're going to reopen the leaderboards and spike in additional training data. For the real tumors, we didn't have any feedback for groups on how they did. So we're going to take 10 or 20% of the validation data, put that on the leaderboard, and invite groups, if they wish, to try to reparameterize and optimize. And we'll leave that open for 2 months and see if groups are able to improve their algorithms using that feedback. And in particular, we'll ask groups to try to designate what they think is the best prediction. What is the one that they think would do best, so we can sort of see if the parameterization insight they have is accurate. In April, we're going to launch a sub-challenge around meta pipelines. We'll make the specific predictions, the VCF formatted files from all of the groups that have submitted available, and we'll ask groups if they can find ways to combine those that would yield more accurate predictions than any group did on their own. This is kind of the wisdom of the crowds, but we're outsourcing the wisdom of the crowds to the crowd. And we think that that would be really effective in drawing in people who don't work on somatic mutation calling, but are really good machine learning people, like a lot of people in this audience. And then, of course, we'll continue with the ongoing releases starting in June or July, I guess, of an in silico tumor 6, and keep those going every 6 months with leaderboards open and available. I'll close by thanking the challenge organizing team. I think I managed to say we all the time. We means a whole group of people. I should really note Josh Stewart and Adam Margolin, who co-led the challenge with me. Every figure that you saw was made by Katie Houlihan, who's actually an undergrad in my team. And you'll hear this afternoon another undergrad, Greg Chen, talk about his work in the AML challenge. And of course, our many data contributors and funding partners who really made this possible. And I'll lastly close by pointing out the winners and how different groups did. And these are the winners for the first 4 sub-challenges. As you can see, Broad SMC won the SNVs for the first 2, WashU for the 3rd, and Broad SMC for the 4th. And then in the structural variations, Delhi team, and then the last 3, Ken Chen's lab, won those. And so I'd like to invite the winning groups on stage for the awards presentation.